everyone. So in this video I'm going to explain how to determine the shrinkage limit of a given soil sample. Shrinkage limit of a given soil sample. So the reference code for determining the shrinkage limit is IS 2720 part 6 from Indian Standards 2720 part 6. We can determine the shrinkage limit of the soil sample. So coming to apparatus required for shrinkage limit is a shrinkage dish, evaporating dish, 425 micron ISC, a balance, and an oven, a spatula, mercury, glass cup, and a glass plate. So these are the apparatus required for determining the shrinkage limit of the soil sample. So coming to procedure, how we are going to determine the shrinkage limit of the soil sample. First of all, we have to take 100 grams of soil sample which should pass through 425 micron ISC. Our first step is we have to take 100 grams of soil sample. This soil sample must pass through 425 micron ISC. After passing the soil sample to 425 micron ISC, we are taking the soil sample first, determining the shrinkage limit. We have to place 30 grams of above soil sample in evaporating dish and thoroughly mix with distilled water and make a creamy paste. Out of 100 grams of soil sample, we have to take only 30 grams of sample in an evaporating dish. Take 30 grams of soil in evaporating dish and mixed with distilled water and mixed with distilled water to form a creamy paste. This is our second step. Coming to third step, we have to coat the inside of the shrinkage dish with a thin layer of grease to prevent the soil sticking to the dish. So before we are placing the soil sample in evaporating dish, we have to coat the evaporating dish. The evaporating dish can also be known as a shrinkage dish. Okay, we have to coat inside of the shrinkage dish with a thin layer of grease so that the soil don't stick to the shrinkage dish or evaporating dish. We have to coat the evaporating dish with a thin layer of grease to avoid sticking of soil to dish. Our fourth step, we have to place the soil paste in evaporating dish and top the dish to eliminate entrapped air. We have to place the sample in dish. We have to place the sample in the dish and we have to cover the top of the dish to eliminate the entrapped air, and cover the top of the dish to eliminate entrapped air. Our fifth step is we have to remove the excess soil from the dish by a moist straightedge and weigh the dish with the wet soil in it. Next, after placing the soil sample in the dish and we have to cover the top of the dish, we have to remove the excess soil from the dish. If there is any excess soil from the dish, we have to remove the excess soil from the dish and we have to weigh the dish with wet soil. We have to take the weight of the dish along with the wet soil which is present in the evaporating dish. Allow the soil in the dish to dry under a fan for about 30 minutes until the color of the soil changes from dark to light. We have to allow the soil to dry for about 30 minutes. Up to 30 minutes we have to allow the soil sample to dry. We have to see whether the color of the soil changes from dark to light color. Until the color of the soil changes from dark to light color, we have to dry the soil sample under the fan. We have to keep the dish with the soil in electrical oven at a temperature of 105 degrees to 110 degrees centigrade for a period of 10 to 24 hours. Next is we have to keep the dish with the soil sample in an electrical oven. In an oven, we have to keep the dish along with the soil sample in an oven at a temperature of 105 to 110 degrees centigrade for a period of about 10 to 24 hours. Our next step is we have to determine the volume of dry soil pad by displacement of mercury. We have to determine the volume of dry soil pad. We have to determine the volume of the dry soil by displacement of mercury. By displacing the soil sample with the mercury, we have to determine the volume of the dry volume of the soil sample. Fill the glass cup completely with mercury and place the glass plate with prongs over the cup. Wipe out excess mercury. Place this arrangement in evaporating dish. Remove the glass plate. Our next step is we have to fill the glass cup completely with water. We have to fill the glass cup completely with the water and place the glass plate with prongs over the cup. And we have to place this cup, we have to fill the glass plate completely with water and we have to place the glass plate over the cup. Wipe out excess mercury. Wipe excess mercury. If there is any excess of mercury, we have to wipe or clean the excess mercury. Place this arrangement in evaporating dish. Next step is we have to place this arrangement in an evaporating dish. Now place the oven-day soil sample on the top of the mercury and press it into mercury with the prongs of the plate, then remove the arrangement from the evaporating dish. Next we have to keep the oven-dried soil sample over the top of the mercury. Place the oven-dried sample over this mercury, over the top of the mercury. On the top of the mercury, we are placing the oven-dried soil sample what we have prepared before, and we have to press it into mercury with the prongs of the plate, and we have to press the sample into the mercury by using prongs. Weigh the evaporating dish with displaced mercury. Our final step, we have to take the weight of the evaporating dish with displaced mercury. Means in evaporating dish, how much mercury is displaced. We can take that weight as a final weight. So once again I am going to explain the procedure how to determine the shrinkage limit. First of all, we have to take some 100 grams of soil sample from a thoroughly mixed portion of material which must pass to 425 micron ISC. Next, out of this 100 grams of soil sample, we have to take only 30 grams of soil sample in evaporating dish, and we have to thoroughly mix it with distilled water so that to make a uniform or a creamy paste. Next, we have to coat the inside of the evaporating dish or a shrinkage dish with a thin layer of grease to prevent the soil from sticking to the dish. Next, we have to place the soil paste in an evaporating dish and we have to cover the top of the dish to eliminate the entrapped air. Next, we have to remove excess soil from the dish by using a straight edge, and we have to weigh the dish along with the wet soil. Next, we have to allow the soil in the dish to dry for 30 minutes under a fan. We have to dry the soil sample until the color of the soil become dark to light color. We have to keep the dish along with the soil sample in electrical oven at a temperature of 105 degrees centigrade to 110 degrees centigrade for a period of about 10 to 24 hours. Next is we have to determine the volume of the dry soil pad by displacement of mercury. Next, we have to fill the glass cup completely with mercury. We have to fill the glass cup completely with mercury and we have to place the glass plate over the cup, and we have to wipe off excess mercury. If there is an excess mercury, we have to wipe off that excess mercury. Now we have to place the oven ray soil sample over the top of the mercury, and we have to press the soil sample into mercury by using the prongs. Next we have to remove the arrangement from the evaporating dish. Next we have to weigh the evaporating dish with displaced mercury. Hope you like this video. Please subscribe to my YouTube channel and share to friends. Thank you everyone.